I'm coming from the Laboratory of Electron Microscopy. We are core facility, and our aim is to provide our users with the best possible images and data for their work, as well as to make our life as easy as possible, right? So I'm coming from Czech Republic. It is in the middle of the Europe, just next to the Germany and Austria. We are located in South Bohemia. So if you know the Bohemian lifestyle, that's us. And the city we are based in is České Budějovice, or Budweis in German. If you think you never heard of it, you are probably right, but I'm pretty sure that you heard about our most famous product, and that's the Budweiser beer. If you didn't encounter the original Czech one, you probably saw or tasted the American copy. Yeah, I said we are the Laboratory of Electron Microscopy. We are based or affiliated to Institute of Parasitology and Biology Center of the Czech Academy of Sciences.
If you are showing your slide, we are not seeing it.
We are not
sharing?
seeing.
Yeah.
Yeah. Well,
because at least I am not. Are you sharing your screen?
Yeah. No.
Probably it's on a different monitor.
Share. Okay. So now, do you see anything?
It's coming.
Yeah.
Good. Okay. Yeah.
That was the first slide.
Yeah. Okay. Good. All right.
Do you see it?
Yes.
Good.
Sorry to interrupt.
Yeah. So that's the Czech Republic next to the Germany. That's South Bohemia, and that's Budweis, České Budějovice, and that's the beer. Yeah. So now you see, maybe it was funny. So we are the Biology Center. We are part of the Czech Bioimaging and Euro Bioimaging network. So most of our users are coming from the campus, but we are collaborating with people basically worldwide. And we have 4 electron microscopes, to transmission to scanning electron microscopes. We cover a broad range of methods from single particle through cryo, all the way to volume electron microscopy. The stereo block phase and array tomography are highlighted here because that's what I do specialize at. So I am working mostly with these two. We organize many courses and events. So I want to show you the 2 EMBO courses that will be happening here this year, 2026. The first one, it's more practical on sample prep and stuff around that. It will be for the 5th or 6th time here. And the other one, it's brand new. It's focused on software. We put it together with Ilya Belevich from Finland, Michelle Darrow from UK, and Kedar Narayan from the US. We'll be focusing on the processing, so mostly we will use Napari, Microscope Image Browser, and Amira. And the first one is already on the EMBO web pages. The other one should be there soon, so you can look it up and find the details if you are interested. What we found when we are interacting with our users, that the ultrastructure is no longer enough. That people are asking less about the ultrastructure, but more about the context. Like, what is that organelle? What is it made of? If I feed some compound to my cells, where did it go within the cell? We had one study that we combine a lot of methods. So basically, this is the diethylamide protist. It's a sea living unicellular organism, and we found that there are huge crystals in its cells. And when we use the EDX and Raman analysis, we found that these are actually the strontium barium crystals. So the strontium barium is actually present in the seawater, but in really low concentrations. So it's actually interesting that some creature it has to be a lot of energy and specialized machinery to pump it out and enrich it, so there can be these huge crystals. So we still don't know what would be the purpose. But it was kind of the case study, and based on that, we acquire a new instrument which combines high resolution SEM with Raman platform. So this can be standalone, but we can put it on the chamber and use Raman directly within the chamber. We have the flat quad detector, so EDX, one of the most sensitive at the market now. So we can use low energies. We can see the low elements. There is the cathodoluminescent detector, which we can detect different wavelengths for the cathodoluminescence, and it's in full cryo. So this system is really unique. You can find different combinations, but with most of it, we heard there might be one system like that in US, one in Japan, but we don't know any other details about that. Are there still newer organelles and structures out there? And I think that many people that are studying the human tissues and stuff, they think there may not be. But the fact is that there is actually a lot of vesicles that we still have no idea about, right? But there are also some creatures that we didn't see before. And this is, again, one of the diplonemes. And when we were reconstructing its cells, so it's studied here at the Institute, you can see that there were like some weird organelles, some huge tubules, concentric structures of the membranes. And the hypothesis was that this organelle is involved in digesting the food because it was close to the endocytic entry point for the cell. So how we can prove that it is actually part of the endocytic pathway? So we can feed the cells with some tracers that we can then later find, right? In the endocytic field, people are using the nanogold particles, but when we try to use the nanogold, it did precipitate in the seawater. But at that time, we were using also the lanthanum for other experiments, so we tried to synthesize the lanthanum nanoparticles, we fed them with the cell, and yeah, they actually internalized them, and we could trace where these goes. And they also went to these organelles and to a lot of vesicles around. So these are the TM images. These are the data from the array tomography. So you can see that actually really get into those new organelles as well as to the main vacuole of the cell. We do a lot of immunolabeling, and so we asked, like, can it be improved? Can we label more epitopes? I think if you do some immunolabeling, you know that what you can use is you can use different antibodies with different size of the nanoparticles. So for example, 10 or 15 like that. But what we try to do is to increase or double the labeling number by labeling the top as the bottom of from the grid. It was published in these two papers. You can't really use the transmission electron microscope for imaging these, but you can use the scanning electron microscope. And the way how you can do it is you can use the STEM detector with high voltage, so it actually detects the same way as the transmission electron microscope what goes through. You can see the image here. I'm not sure what the quality you see, but there are nanogold particles. And then you can switch and use the PSA just with low voltage to see what's on top. So that's this here. So you can see that you don't see all. And when you subtract the top particles from the rest, you see which one is actually on the bottom. The drawback here is still that although now the microscopes are really good, really well aligned, but there is still some shift when you change from 30 to 1.5 kVs. Plus you need to scan it twice, so the dose is higher. So what we found out actually that you can actually use just one voltage to see both particles and distinguish them. So when you use too high voltages, yeah, everything goes through and you don't see the difference. If you use too low voltage, you see just the top, or the resolution not really good. But if you use something in the middle, so the exact, it depends on the thickness of your section that you are looking at, but the details can be found in the paper. You can see that you can actually see two classes of the particles, one that are like more bright and one that are more blurred. So that's the difference. The top one would be brighter and the bottom one would be blur, because there is more scattering when they go through the sections and back to the detector. And yeah, if you are using different sizes, then you would see the different population as well. Now we are working also how to distinguish them with the software and stuff. So now I'm getting to the paper that is actually in the main title of my talk, and it's the minimal resin embedding posterior block phase samples, and now we can help you with finding region of interest and the charging. So for those from you that are not familiar with the volume, SCM, or serial block phase, the principle is that you embed your sample in the resin as you would do for the transmission electron microscope. Then you put the sample inside of the SCM, where is actually the ultramicrotom. So there is a diamond knife. You imaged your block of the sample. Then you cut the section off. You image it again, cut another section. And then that way you end up with stack of the images that you can then analyze and get the information about your 3D volume. Our aims, or my aims for serial block phase, is always to have the best resolution possible, image the largest volume possible. You need to find your region of interest, and against that goes always the imaging time or cost, because obviously the higher or lower pixel voxel size or largest volume, the longer it will take. And also we struggle with the charging. This is how it looks. So this is the typical serial block phase sample. It's some cube of the resin that you put inside, and you can't really tell what's in there, right? And the charging, if your sample charges up, it efficiently blocks your imaging. There are ways around it. I will show you some of them. So how to find your region of interest in the serial block phase? You can use the correlative approaches or micro CT. Here I am showing how we did use the fluorescent to find Borrelia inside of the tick. Or you can use the serial block phase as an expensive microtom and just slice through it and wait until your region of interest appears and then start imaging. But it can take a long time, I mean days or weeks. But then there are samples that we needed to image, and we were interested in the structures on the surface. So first was the micropyle of the fish roe. So that's the entry point of the sperm inside of the egg, and also the colophore. That's the excretory organ of a springtail. So the excretory, that's the organ, that's my co-author on the paper studies. And we tried to use the minimal resin. The minimal resin method was actually published for the FIPSEM by Nicole Schieber and the group of Yannick Schwab in the EMBO Heidelberg. And what they did, they actually infiltrated the resin in the sample, and they get rid of the extra resin. So when they put the sample later on in the FIP, they could actually see nicely the surface. So you can see that in zebrafish, they can pick which septum they want to dig the trench in. For C. elegans, you can also navigate into the part of the body that you want to image. Image is very good. So we try to adapt it for the serial block phase. And here are the serial block phase samples. As you can see that for the micropyle, we really see nice details on the surface of the fish roe. And for the springtail, this is actually the colophore, the excretory organ that we are aiming at and what we want to image. So here is the comparison. If you prepare the sample the traditional method, you can see that it's hidden in the resin. You have no idea where you are. You just blindly go in. But if you use the minimal resin, you will see that this is your region of interest. What is nice that you can actually image it from the side and measure precisely the amount of material that you want to trim and get rid of before putting it in the serial block phase. So that's what we did. So you can see that here it was almost a 0.5 millimeter thing. It's always good to leave some material above your region of interest, so you can align the knife and set the imaging parameters first, and then you start image what you want and what you need. So what about the charging? So I showed you this is the typical sample, and if you have a lot of resin, it typically charges. And it insulates also the sample that otherwise would be cut. So these are some examples how the charging can look like. Can be like these blurs at the places where the resin, that is just a resin that looks innocent unless it's in part of the cell or disrupting the cell like here. But it's bad when you want to do the image processing. So for the alignment and these kind of stitching, it can be a problem because for the software, it doesn't tell it apart from the signal. So for it, it is a signal. So there can be problems later on. So there are solutions how to deal with that. So you can lower the dose, but it's not always possible because if you lower the dose, you lower the signal, and it's then hard to image anything. The most common is actually the variable pressure. So what we also use, that you actually put some gas inside of the chamber. The gas molecules get ionized, and the ions then removes the charge from your sample. And there is also this focal charge compensation device, which is part of the Zeiss machines because Zeiss have patented it. And it's actually putting the gas only on your sample, so it's not in the whole chamber. So it has much better resolution than, because the variable pressure decreases the resolution it's a little bit. But yeah, you can also only use it with the Zeiss. We are using the thermo machine, so it's not for us. You can play with a sample prep. And there were some attempts with using the conductive resins. So this is the example. This is work of this Japanese group. What they did use is the carbon black filler, the Katyn black. They put it into the resin. And you can see it here that it's basically surrounds your tissue, but it doesn't enter it unless there are some cavities. And you can see that it really helps you dissipate the charging. So without the carbon in there, you have the charging. And if you add the carbon, it helps the charge to dissipate. And here you can also see that the quality of the imaging is actually much better. There is this another example, that's work of Christelle Genoux on zebrafish and on Platanaris worm. And they actually use the minimal resin, but before imaging, they cover the sample in the silver epoxy, as you can see it here, to increase the conductivity and also the stability of the sample. What we are showing in this paper, the minimal resin one, that you actually don't need to cover the sample with anything. It's just get rid of the extra resin and simple sputter coat the sample is enough. So you can see it here. If there is the extra resin around, it insulates the sample. So if you zoom in and try to image in details, you can't really do that. While if you get rid of the resin, you really can image in high quality, better doses. You have more signal. You can generally image in the high vacuum mode, which wouldn't be otherwise possible. So these are like the different samples of different specimens that we are using. So you can see that there is always some resin around, depends how you prepare it. And usually when it's not that much, it doesn't really matter. When you have a little bit more and the sample is not as great, then you still need to image in the low vacuum. So it can be like, but it still improves. So it can be still like 10, 20 pascals compared to like 40 or 50 that you may need to use without removing the extra resin. So to go quickly through the protocol, you take your sample, you process it as you normally would do it, and you put it when you are in 100% resin. Before the polymerization, you actually remove the excess of the resin. So you put it on the paper. And for the FIPSEM work, they actually use the halogen lamps to heat the sample, make the resin more viscous or less viscous, that it would drop out. What we actually dip it into the acetone to dissolve the extra resin. And then when you are happy with your sample, you put it on the pin and polymerize it. This is just to show the difference when without the acetone and with the sample that was wished with the acetone. So without the acetone, generally it's good unless you need to find some super nice structures like the micropyle, right? So for these ones, we really needed it. At first, I was really afraid that I would dissolve the whole sample But actually, I was surprised that the acetone was not that aggressive, that you actually, if you just dip it in there and take it out, it's not enough, because the 100% resin is really thick and it's hard to dissolve. So you really need to put it there, vigorously shake it for quite a long time, like tens of seconds or even a minute. And then you take it out and you get this kind of the images. So the stability and support is always important. So what you aim and need, that the base of your sample is always wider than the top, so you won't have the chatter. So when the sample was thin and narrow, or it was tiny, that we were afraid that we may hit the pin with the knife, then we provided some extra resin support for them. And we always cover the base with a colloidal silver. So it helps with the stability, and it also covers all the crevices. So when you do the sputter coating later on, it's uniform and the charge can dissipate. And because of this approach, the imaging was actually that great that we were able to use the multiple energy imaging. So it's available on Thermo Fisher machines. I'm not sure if it's patented, but the other suppliers don't have it, so it may be patented. So the thing is that you having the physical sections, so you cut the section, and when you are imaging your block, you can use different KVs to get deeper into your sample. So this would be the 1 energy, 2nd energy, 3rd energy, and then the software deconvolutes and you get higher resolution imaging. So in this case, we were cutting 50 nanometer sections. I was using energy for 25 nanometers and 50. But some samples were that great that I was actually able to get to isometric voxels, so 10 times 10 times 10 nanometers. So that's almost close to the FIP. Here it's even better, 8 times 8 times 10 nanometers. So in this case, I was cutting 40 nanometer sections, physical sections, and then using 4 energies. So it was this final resolution. So I hope that I showed you that for certain samples, the minimum resin is great, and it can really help you to find your region of interest and to help with the charging. And I think I still have a couple of minutes. So I want to show you our latest paper that was published in Nature Communications in October. It was even listed among the 50 top about the microbiology and infectious diseases. And we wanted to see the Borrelia bacteria in the skin. So the Borrelia is the source of borreliosis, is the disease transmitted by the ticks. So as mosquitoes do transmit malaria, the ticks do transmit their own diseases, for example, Borrelia. So the source is actually the bacteria, the spirochete bacteria. And when the tick bites and sucks the blood out of you, the Borrelia can get into your skin. That's not really the problem. The problem is when these bacteria get into the joints, they can get into the heart, they can get into the brain, and that's where the disease can be a big problem. But until now, no one really knew how the Borrelia moves in the body, in the tissues, how they can get from the tick bite site to the joint, for example. So we tried to simulate the tick bite. We injected the Borrelia into the skin of the mouse, take the biopsies, and look at what the Borrelia do in your skin. Turns out that they are entering the lymphatic system pretty soon on. Here you can see it inside of the lymphatic capillary, and it actually enters the cells that it makes invagination into the lymphatic endothelial membrane. You can see it here. It can then get through the cells. It is a bit different because then we wanted to see if they can also get to the blood capillaries. It turns out that the blood capillaries are much harder to get into because they are more structural rigid. And what was really interesting to see, because there are the endothelial cells that cover the whole volume, and then you have the specialized pericyte cells that do play a role in a control of the vasculature in general. And the pericytes cover just smaller portion of the vessels. But the borrelia were 50% time attached to these pericytes. So it looks like the pericytes really play some role in its attachment to the bodies. So I want to there's the early disseminations that it looks like that the borrelia doesn't really damage the tissue. It's probing for the weakest point where it can go further. So here it really looks like the base membrane can be a big barrier for them. So they are looking around the pericyte where can they cross it, or that barrier is not present in the lymphatic passage and they are freely going through that. That will be all. I would like to thank all our collaborators and all members of our lab, because without the teamwork here I wouldn't be able to show you anything. And also thank you for your attention.
Thank you, Jiří. This is great talk. Any questions from the audience? Let me see. I have a question. First, about the double-sided immunogold labeling. And how do you do it? When you do the double side, you just floating it on the surface and then flip it, or you have other tricks to make sure that you don't have the crossover?
Yeah, either you float it, but to save, I think, the volume, what we do, we have it in the tweezers and just add a little bit, so you can use smaller volume. So you stain it in the tweezers.
Wow. Okay.
So that's really just a couple of microliters.
So you do it manually, you hold tweezers holding the grease.
Well, you are using the tweezers that hold the sample. I mean, the one that you that you need to press to open, right? So it holds the grid the whole time. You don't need to.
Oh, okay, all right.
Yeah, I don't know how it's called in English.
Yeah. Okay, great work. Also, the when you do the minimum resin with the serial block face, does the knife have problem to section these irregular shapes? You don't have any problem?
No, it doesn't really matter.
Okay.
Because I tend to, when I have the square or there is the line, I tend to align it with the knife. But I think that's really important when you try to collect the sections. But here we are imaging the block face, and then we don't really care how it is. So it's just a matter of habit to aligning the sample, the edge with the knife, that we get all when we start sectioning for the TM or something.
Okay. But, we have a raise hand. Alexi, you want to ask your questions? Several people are waiting.
Yes, thank you, Jiří. So, hi, Jiří. This is great talk. Very interesting data and very good approach. I like it. But I want to ask you about the first part when you showed combining your SBEM experiment with immunostaining. With the different immunogold particles or a combination of different sizes. So my question is, did you try CLAM or correlative light and electron microscopy? Because I'm sure, because we already started about 5 years ago, and trust me, it's not too difficult. It's pretty easy and more informative, because you have depending on your microscope, you have 5 colors. And combining with the SBEM, it will be a very good data.
Yeah. I mean, it depends what you want to see and how you want to see it, right? Because, for example, like here, the immunogold on the TM sample can give you much higher resolution. So if you are chasing, for example, we had projects that wanted to see that they know that the protein is inside of the membrane, but they wanted to know whether it's facing inwards or outwards. So I'm not sure how you can do it with CLEM. I mean, you can do it with if you have some super resolution ones, right? But otherwise, I think it would be much more difficult.
We have Zeiss Eriscan.
Yeah, but Eriscan is still like the resolution is...
It's pretty good.
50 nanometers or something. What would it, as far as I remember, the new generation is probably better.
It's enough to identify the very small synapses in the mouse brain.
Yeah. Yeah. So. But can you tell, like, for example, I mean, it is possible. I mean, it depends what you use and what's your question or what you aim for. I'm not saying that that you shouldn't use CLEM. I think this is a possibility. It may work. And it worked for us. Yeah.
Do you have
and the thing is that at the moment, like in the campus, we don't have the super resolution capabilities. We need to go somewhere else. So that's kind of a drawback. But hopefully that will change.
Okay.
But, yeah.
Good luck.
Thanks.
Okay. Chris, will you, before Alice, make it quick?
Okay, right.
Sorry.
No, it was just, hi, Jiří. Well, nice talk.
Yeah.
Just really interested in the multiple energy thing offered by Thermo Fisher. What's the best resolution you can get on that? And I presume it's because of computer interpolation of the many energy levels.
Yeah. I mean, if you are not really limited, you can use as many energies as you want, but you are limited by the electron dose still.
Yeah.
Right?
Does it cook the surface?
Yes. So we are limited with the electron dose in any serial block phase.
Yeah.
I was told that if you want to section, like the thin section, talk about 30, 40 nanometers, you shouldn't exceed 25 electrons per nanometer square, something like that. If you are cutting thicker, I know that I can use 40 or even 60 electrons. But for these, basically each energy, it's a sum. Then you need to sum it for the final physical cut, right? So these, I was really using like something like 5 electrons per one scanning. So the combination was then 20. Therefore, I can use the 4 energies. So then it's really down to your sample. If the sample is really great and you can use low energies, then you can use more energies. Does it make sense?
Yeah.
Yeah. But if you are forced to use already 30 electrons per one scanning, then you can't really use more energies. So that is this limitation.
Thank you.
Okay, last question. Alice, quick.
Yeah, very good talk. Regarding, I continue with Ruqing's question regarding the double side of the grids immunolabeling, right? You know that manipulate of the grids will be a little work. And also when you do double-sided immunolabeling, obviously you cannot have a membrane on the grids. So means you have a barrier section on the grids. So after you do so many labeling, what is a successful rate for you to get the section will not be broken? Another thing is, you typically what kind of mesh macro the grids that you are using in order to make sure you have a large enough area will be exposed to make sure the grids will not broken during this whole incubation time.
Yeah. Okay. Like this kind of testing was done by my colleagues, so I can't tell you my experience with that. I was doing more like the imaging and stuff, but it's generally like here. Yeah, as you said, we can't use the form var or something. So we tend to use the lower mesh or more grids. I don't know what's lower, higher. So the higher frequency. And yeah, there is the higher chance that it will block some place that you want to see. But that would be the drawback of that one, the trade-off. But if it depends what you are looking at. If you're looking something that's really rare, then it will be a problem. If you're looking for something that is more common, then it's not that big problem.